Hi, Gen Bio. Dr. Fry here, and this week we are talking about osmoregulation. So, osmoregulation is the process by which our bodies control the amounts of salts, fluids, and waste products that leave our body or are retained in our body. Okay. So this happens by the kidneys. So I'm just going to direct your attention to the picture here. We've got two kidneys right here. There's this big blood vessel. This is the aorta, and it has a branch that goes to each of the kidneys called the renal artery. And then, of course, once the blood travels through smaller vessels through the kidneys, it goes back through the renal veins then to be taken back to the heart. Okay. So I'll also mention before we move on that there's some other anatomy involved in osmoregulation besides the kidneys, although they are the primary organs responsible. There's some other structures. So you've got the kidneys, which lead to these tubes called the ureters, okay, which lead to the bladder, which leads to the urethra. And of course, then you know how your waste products exit your body. So the roles of the kidneys are to, as I had mentioned, eliminate metabolic waste products. So this is waste products that come from the breakdowns of proteins and fats. We also need to retain essential molecules. Some salts will be retained, various ions. And then one other major role of the kidneys is retention of water. So when our kidneys are filtering our blood, okay, the blood's going to be pushed out into the kidneys. A lot of fluid, a lot of water is initially processed, but then taken back up by the body. Okay, so water retention is a huge part of what happens in the kidneys. And then there's also, as I mentioned, some reabsorption of ions. This is done in a controlled fashion. So based on what the osmolarity of our blood is and based on what we need to counteract the osmolarity right at that moment, our body controls for the amount of ions that exit or are retained in our body. Okay. And then pH maintenance is also a role here that happens in the kidney. So we want to maintain the pH of the blood. Okay. So I'll just mention before I move on that waste products, you might hear me mention urea, creatinine. These are from breakdown products. These are breakdown products from protein digestion. And then ketone bodies are going to be breakdown products from fat digestion. Okay, so you might hear me say those as we move through the presentation. Okay, so I'm going to move on to the next slide. You saw the picture of the human body and where the kidneys are located. If you look at an enlarged picture then of the kidney, it looks something like this. And the outer part of the kidney is called the cortex. The inner part is called the medulla. And then that all leads to the renal pelvis, which is right here. Okay. And then if you take a section out of the kidney and enlarge that, then what we're showing you here are nephrons. These little tan yellow structures here are the nephrons, and they're about 1 million that make up each kidney, most of which are cortical nephrons. So the nephrons that stay in the cortex or the outer part of the kidney. Some, however, are called juxtamedullary nephrons, okay, and they extend down into the medulla. Juxtamedullary, they go into the medulla, right? And then they'll go back out and then they connect to what's called the collecting duct. So we're going to take a closer look at those in the next slide, okay? So this is a picture of a nephron. I have it a little bit larger on the next slide, so let's look at that. Okay, so this is a juxtamedullary nephron. And these are largely responsible for maintaining the blood osmolarity that is able to be more dilute. So our blood can retain water and excrete then urine with a higher osmolarity, okay, to the rest of our body fluids. And like I said, the juxtamedullary nephrons are largely responsible for those, as we'll see in the next few slides. But first, a little bit of anatomy of the nephron itself. Okay, so here is the first part of the nephron. This is called Bowman's capsule, this little tan cup-shaped structure, okay? And it surrounds a ball of blood vessels called a glomerulus, okay? So Bowman's capsule sitting right around that glomerulus, okay? See all the little ball of blood vessels. And that came from the renal artery. The afferent arteriole is this little segment right here. The renal artery, as you saw in this picture here, is the larger blood vessel, leads to the afferent arteriole, and then that leads to the glomerulus, okay? And so blood is flowing through the glomerulus, and the walls of these blood vessels here are leaky. And as blood pressure is exerting a little bit of force on the vessels, a lot of the fluid is being pushed out into Bowman's capsule, okay, the first part of the nephron. So you've got a lot of fluid now that moves from Bowman's capsule into the proximal tubule, which is right here. This is the next part of the nephron. And then down into what we call the loop of Henle. Okay, and the loop of Henle has a descending and an ascending limb. And then that leads to the distal tubule and the collecting duct. And then it'll go and exit out the kidney through the renal pelvis. Okay, so as that pressure from the blood is pushing the fluid out into Bowman's capsule, now once it's out of the blood vessel, we call it ultrafiltrate, okay? Which just means it's a lot of the liquid part of what was in the blood, but it needs to be processed before we can release it as urine, because we want to keep a lot of the stuff that's in that ultrafiltrate, okay? We want to keep a lot of that. So it needs to go through a lot of processing before we can release whatever waste our body does not need in the urine, okay? So there's a lot of selective reabsorption of solute molecules, a lot of retention of water that takes place along this process. We will talk about all of that in the next couple of slides, but let me just recap a little bit, just sort of map out the pathway of filtrate is going from Bowman's capsule to the proximal tubule, the descending and then the ascending limbs of the loop of Henle, then the distal tubule, and then the collecting duct, and then into the renal pelvis, which you can see here, down the ureter, to the bladder, to the urethra, and out the body, okay? All right. This is kind of also just a recap, just spelled out in case you missed anything. Blood pressure is forcing that fluid into the lumen, the space in Bowman's capsule, and this fluid includes all sorts of stuff. Like I said, a lot of that we actually want back. It's starting to leave the body, but we have to say, wait, we want some of that back. Hang on, which we'll get to. But a lot of that fluid at that point has salts, glucose, amino acids, vitamins, nitrogenous waste products such as urea, other small molecules. Obviously included in the salts is sodium, chloride, potassium, and the nephron itself is lined by a transport epithelium, an epithelial layer that is responsible for moving the filtrate either across back into the body or back into then the nephron so that it will ultimately leave the body, okay? Like I said before, one of the most important roles of this epithelium is reabsorption of water, okay? Another important role is reabsorption of salts if we need them. And ultimate goal is to maintain homeostasis. We need to maintain our necessary degree of water in the blood. We need to maintain the amount of salts in the blood, and we need to maintain the pH, okay? So for example, if you haven't been drinking a lot of water, or if you were sweating a lot, if you were exercising, or other reasons you might be dehydrated, eating a lot of salty food, your body is going to want to reabsorb a lot of that water. So it's going to produce a lower volume of urine. You're still going to produce urine, but the volume is going to be less because you've reabsorbed much of the water that had initially exited as ultrafiltrate. Okay. All right. So now let's look at how this actually happens in the nephron. This is, again, a juxtamedullary nephron. Okay. We've got Bowman's capsule. We've got the glomerulus. As the filtrate moves through the proximal tubule, you've got a lot of things being reabsorbed, either actively or passively, right off the bat. So you can see there's some active movement of salts, hydrogen, some passive movement of water, some passive movement of ammonia and bicarbonate. These things are going to be automatically reabsorbed or excreted into the rest of the nephron. Okay. Now I'll mention that as we go and move into the loop of Henle, the descending limb of the loop of Henle has almost no channels for salts. And as we move further into the medulla of the kidney, the interstitial fluid, so the solution and the solution surrounding the nephron is getting increasingly osmotic, so that it's got a higher osmolarity in the medulla than it does in the cortex. So as the filtrate moves down the descending limb, water, which can exit through the descending limb, is going to be moving out into the surrounding tissue. And it's going to be mostly passive because, like I said, this medulla, the medulla is very highly concentrated. The interstitial fluid has a high concentration of solutes. And so just because of osmosis, water will passively move out, okay? And more so the further into the medulla the loop of Henle goes, okay? And then as the filtrate rounds the corner here and then moves into the ascending limb, now this portion does not have a lot of channels for water, but instead has channels for salts, for solutes. So there's going to be a little bit of passive movement, just again because of osmosis, of salt now, because there had been a lot of water exiting here. Now, to even that out, just because of osmosis, we're getting movement of salts, okay? And first this is passive, but as we go further up towards the cortex, then it's going to be an active process, okay, because of just the osmotic pressure, where now it's going to be an active process. And then as the filtrate moves into the distal tubule and the collecting duct, here we get some regulation based on the actions of a few hormones. So we're going to talk about those hormones on the next slide. But these are acting to either retain water or retain salts, or a combination of the two, okay? So salts, waters are going to be retained if necessary based on what the body is sensing, what the body knows is going on, either regarding osmolarity of the blood, based on some receptors that are able to sense for the osmolarity of the blood, or receptors that are sensing the pressure of blood. Okay, so all of this is well sensed by various receptors and then responded to by various hormones. Okay, so let's talk about those hormones. So the hormones that we're going to look at are anti-diuretic hormone, which is also known as vasopressin, and then the Rast system. The Rast system involves renin, angiotensin, and aldosterone, as well as a few intermediaries. Again, we'll talk about those. But let's talk about anti-diuretic hormone first. This is a hormone produced in the hypothalamus, which is in the brain. Okay, and I have a picture of it here. Hypothalamus is right here. This is, of course, your head. your brain, hypothalamus, and just below the hypothalamus is the pituitary gland. So anti-diuretic hormone is actually produced in the hypothalamus and then released from the posterior pituitary, which is just below it. And the pituitary, you may know already, is responsible for a lot of hormone release that goes throughout the body. Okay. So one of these hormones is anti-diuretic hormone. And the release of this is stimulated by an increased blood osmolarity. So the body senses that there's been an increase in the osmolarity of the blood, meaning you've got a lot of salts in your blood or you don't have a lot of water in your blood. So you either haven't drank for a while, you've been sweating a lot, eating a lot of salty foods, etc. So you've got this increase in blood osmolarity. And what the body needs to do then is to even that out, right? Decrease the osmolarity. And it does this by reabsorbing water, maintaining as much water as it can. And this will happen in exchange for producing a smaller volume of urine. Okay. So when you are releasing anti-diuretic hormone, this is signaling then the kidneys to reabsorb water. Okay. Don't let that go out as urine. Keep it in the body, okay? And so that's what happens when anti-diuretic hormone is released. Let's look now at the Rast system, okay? So the Rast system is the other mechanism in place to control for the amounts of salts or water that leaves the body as urine, okay? So renin is the first part in this process. It leads to sort of a cascade, a signaling cascade, which we'll talk about on the next slide. But it's activated when there is a decrease in either blood volume or pressure or a decrease in blood sodium. Okay. So for instance, if you just drank a lot of water, ate a bunch of non-salty foods that have maybe yogurts or something that have like a high water content, some kind of soup that doesn't have a lot of salt. If you have a high blood volume that does not have much sodium, so lower sodium, or if you have a decrease in blood volume, which will lead to a decrease in blood pressure, excuse me. Sorry about that. So you could have a decrease in blood volume, which would lead to a decrease in blood pressure. So the lower the volume of the blood, you're also going to, what will accompany that is a decrease in blood pressure or a decrease in blood sodium. Either of these will activate the Rast system, okay? And the goal of this then, this activation, is to promote reabsorption of sodium. So this will counteract the decrease in blood sodium, or if the problem was a decrease in blood volume, which causes a decrease in blood pressure, then if you reabsorb sodium, often what happens then is that chloride and water will passively follow, again, because of electrical attraction between sodium and chloride, and because of osmosis, water will follow solute movement, okay? So either way, you're going to be able to correct the problem. You're going to correct the deficit, okay? All right, so let's move on to the next slide where I talk about this system just a little bit more. There's a little bit more detail involved in this system than with the ADH release. So we have another slide on this. So again, the renin, angiotensin, aldosterone system starts with renin, right? Renin is released from the kidney. And this happens when this structure called the juxtaglomerular apparatus is sensing a decrease in blood pressure or blood volume. Okay, and this happens right here at the kidney, okay, right next to the glomerulus, right, juxtaglomerular apparatus. It's going to sense a decrease if there's a decrease in blood pressure or volume, and then release renin from the kidney. And this is going to cleave angiotensinogen, which is produced in the liver, and that will then convert to angiotensin 1, and that is later converted to angiotensin 2. Angiotensin 2 is the thing that we're gonna see act here then. It's gonna be acting as the hormone, which will constrict the blood vessels first. It does a couple things. First, constricts the blood vessels, which will increase blood pressure. And then it's gonna trigger the release of another hormone, aldosterone. Okay. And this comes from the adrenal gland, which sits right on top of the kidneys. Let me go back and show you that. I have the kidneys here. Adrenal gland sits right on top. Okay. So then you've got the release of aldosterone from the adrenal glands. This will then cause reabsorption of sodium back into the body, and water is going to follow. And this will both increase the blood volume and also the pressure of the blood. Okay. So again, we're counteracting a problem that the body sensed was happening. Okay. And we're trying to basically even that out, maintain homeostasis, right, with both of these systems. Okay. So far we talked about movement of water, which will obviously alter blood volume and pressure. We talked about movement of salts. Okay, so you're going to change the concentration of sodium, chloride, general solute content in the urine versus the blood. The other thing that we need to talk about is pH regulation. So blood pH has to be maintained at a relatively constant level. And one mechanism that helps with this is the sodium hydrogen exchange pump. Okay. So when sodium is moving, say, from the developing urine back into the blood, it happens by way of this pump that just swaps sodium for hydrogen. Okay. So sodium's going in this direction, hydrogen's going in the other direction. Okay. So when we're retaining sodium, as we had said we did with the Rast system, right, promotes the reabsorption of sodium back into the body, what's going to happen then is that hydrogen's going to move the other way. Okay. Sodium moves back in the body. Hydrogen is going to be at a greater concentration in the urine. And you probably are familiar then with the fact that the amount of hydrogen in a solution is a direct indicator of the pH. So the more hydrogen in the urine, the lower the pH is usually going to be. Okay. So when Rast is activated, when that Rast system is activated, you're getting a lot of reabsorption of sodium, just like shown in the picture here, in exchange for hydrogen in the urine. Okay. And this will obviously cause a lower pH. Okay. So those are the factors we're going to look at in lab this week. We're going to look at urine volume. We're going to look at specific gravity, which is a measure of the solutes that are in your urine. We're going to look at chloride concentration. And if you remember, I said chloride is often sort of an indirect indicator of sodium in the urine because it follows sodium, electrical attraction. And then the pH is the fourth thing we're going to look at, okay, when we look at our urine this week. Okay. So if you remember, hopefully you did, that I said that you should try not to urinate, eat, or drink for 2 hours prior to lab. Okay. As soon as you come in, we're going to have you void your urine. We're going to have you drink water. And some of you, if you want to volunteer, feel free, will take a salt tablet. Okay. And then you're going to void your urine every half an hour. And we're going to look at those 4 variables that I just mentioned: urine production in milliliters, how much volume, the pH, the specific gravity, and the chloride content. Okay. This is measured in milliequivalents per liter. And like I said, chloride content is an indirect measure also of sodium in the urine. Okay. And we're going to compile that all on an Excel spreadsheet, and we're going to look at your data and the data from all of the other labs. We like to have a lot of data for this one. And then you are going to analyze the results that we find. Okay, we're going to give you that data, and then you will make averages just like you're doing for this week's lab. You're going to make averages, and then you're going to write your discussion section based on the results that we find. Okay, so let's talk about those results. Just to recap, I'm hoping that throughout the course of the PowerPoint, you kind of got a feel for what results you should expect, but I'm going to lay it all out right here for you. Okay. So the group of students that just drinks water, they're going to have a lower blood osmolarity. So they've drank a lot of water. They hopefully have not ingested much salty food recently. And so what the body is going to do is it's going to want to act to even that out. It's going to want to retain sodium. Okay, so it's going to move that sodium back into the blood. And that's going to happen through activation of that Rast system. Okay, where the kidney is then going to reabsorb sodium. And you have to recall that's going to be in response to a release of hydrogen. And so what we should see is that this group will produce a larger volume of urine, a lower chloride concentration, a lower specific gravity, and a lower pH. Okay, we're obviously seeing more volume of urine because they're not retaining that water. Okay, they're trying to retain the sodium. And then if they retain the sodium, they're also going to retain the chloride. So you're going to see less of that in the urine. You're going to see less sodium and chloride. So there's going to be a lower specific gravity in the urine. And then you should see a lower pH because as sodium is retained, hydrogen is going to be excreted in a greater amount. And as that happens, you should then see a lower pH of the urine. Okay. Now this is, I'm going to stress, in comparison to the other group. Okay. So when we look at the water group, the water only group, compared to the group of students that had drank water and taken a salt tablet, these guys, they've got a relatively higher blood osmolarity compared to this group, right? So the folks who ate salt, they're going to have a higher blood osmolarity, right? This is going to trigger the release, hopefully, of anti-diuretic hormone, ADH, which is going to promote the reabsorption of water. You just ate a lot of salt. We need to fix that. Let's get some more water kept in the body, right? So they should not be releasing aldosterone and not reabsorbing sodium. We don't want that extra sodium to be kept in the blood. We want it released in the urine, right? For this group. So what you should see then is that the group that drank salt or drank water and ate salt would have a lower volume of urine produced because we're keeping as much water as we can. We're going to have a higher chloride concentration because we want to get rid of that, get rid of the sodium, get rid of the chloride in the urine. And so the urine will have a higher chloride concentration. The urine will also have a higher specific gravity. All those solutes, move them out, right? And then they should also have a higher pH because, again, sodium is going to be in the urine in exchange for hydrogen moving back into the interstitial fluid and back into the blood. Okay, so the urine should have less hydrogen ions and therefore a higher pH, okay, numerically. All right, so those are expected results. This is what you're going to base your discussion writing assignment on this week. Again, I can't stress enough, the point is that you're comparing the results between the two groups, not comparing the results in one group from the beginning to the end of the experiment. For example, you haven't peed, some of you, probably all night when you come to class. You may not have peed since you went to bed the night before. Your urine volume is going to be pretty high, regardless of whether you drank salt or drank water and ingested salt. The first time point will probably have the highest urine volume. And then we're going to have you pee every half an hour. So as the experiment progresses, the volume of urine in both groups will probably decrease from time point 0. What we really want to look at, though, is not whether or not it decreases over time, but we want to see if it decreases more compared to the other group or less compared to the other group. So over time, the salt group, they're going to be having some loss of water, but probably not as much loss of water in the urine as the group that drank just water and did not ingest salt. Okay. So again, we're comparing the two groups. Okay. You don't want to really focus on over time, like at the beginning. You want to focus on where at the end of the experiment the two groups lie in relationship to each other for the 4 different parameters: volume, chloride concentration, specific gravity, and pH. Okay. So in the discussion section, you want to discuss trends. You don't need to give me a whole lot of numbers. You don't need to make a graph, although it will help you to make a graph. If you make a graph, you will be able to see your results a lot more clearly. You don't need to give me that graph, but you do need to discuss the results that we saw. So in order to do that, you might want to just make the graph so you can see what happens. And in the discussion section, you also need to demonstrate that you understand the roles of the two hormone systems that are at work here, okay? And then lastly, I have a few common mistakes I want to stress. Often, students will use the word significant. They'll say there was a significant difference seen in the volume or whatever. The word significant in science writing implies statistical significance, meaning that you did a statistical test and you proved, well, you strongly supported your conclusions based on statistical analysis. We are not doing statistical analysis, so you should not use the word significant. It means something else in science writing, okay? Again, I'm kind of gonna beat the horse dead here. Don't focus on whether or not the variable increased or decreased over time. That's really not that important. It is important to compare it to the other group by the end of the experiment. So like around like the last time point, hopefully we'll see that the two groups show different results, okay? All right, that is it. I will see you guys on Tuesday, and hopefully you all are having a good week. Shoot me an email if you have any questions, and I'll see you soon.